Hi, I'm Dr. Amanda Hagee at the University of North Dakota in the Department of Biomedical Sciences. And today I'm going to show you how to check your cell cultures using a basic inverted microscope, just to check them for contamination or confluence. So I've taken my cells out of the incubator here, so I'm already in the necessary PPE for working with human cell lines. It'd be similar if you were also working with mouse cell lines as well. So I am wearing a lab coat. I have long pants and closed-toed shoes on, as well as gloves that I sprayed down with 70% ethanol before touching and putting my hands inside the incubator to get my dish of cells. So when pulling my cells out of the incubator, one of the first things I want to do is look at the condition of the media. So the media should be bright pink red. It should not be yellow. This indicates that the pH here is stable, normal pH for the cells themselves. The color of the media should also be translucent, so there should not be any cloudiness or particles floating in the media. That would possibly indicate that there's a contamination going on. So I can check that by eye first, and then we'll look at the cells underneath the microscope. Again, just with handling any sort of surface, when you're handling cells, you want to make sure it is sterile. So you want to spray down that surface with 70% ethanol. So before I set these on the lab counter, I'm going to spray the counter with ethanol. And then before I set them on the microscope, I'm also going to spray that with ethanol. And then I'm using chem wipes just to wipe the ethanol down. So we're just using a basic inverted microscope here. So it should be covered when not in use. So you can take the cover off here. And an inverted microscope means that the objectives or lenses on the microscope are on the bottom instead of the top. So you can see the different objectives on the rotator here along the bottom of the microscope. To turn it on, there should be a power button here on the side. This one has a little bit of a delay, but you should wait, and then you should see the light coming through the objective. Just like the microscope is stored always with the cover on to prevent dust from getting in the objectives, it should also always be set on the lowest objective possible for those. So here we have it on the lowest or shortest objective. It's usually lined with red compared to the other ones, which are yellow and green here. So it should start at the lowest magnification, the smallest objective. So just like I covered the counter with ethanol, I'm going to spray the stage, again to make sure it stays contaminant free. So just a small bit of 70% ethanol and a chem wipe. And then I'm going to place my dish of cells onto the stage, making sure there's no writing from my notes on the top of the dish in the way of the path of light from the objective. And every time I touch my hair or just my glasses or my face, before I touch that dish of cells, I'm going to spray my hands again with 70% ethanol, making sure that my contact is always sterile with them. Okay. So looking at the cells, then I'm going to have to focus. So this microscope has two focuses, a coarse focus and a fine focus. So usually the coarse is the bigger wheel, so that moves the objectives up and down by like a lot when you spin it, versus the fine focus, which is going to be the smaller wheel, which is just going to have it move up and down very, very slightly. Things. So I want to start at the lowest objective and kind of get within range when you're looking at the cells with the coarse, and then adjust more with the fine. So things I'm looking for here, again, would be anything floating in the media, anything that looks like small clumps of bacteria or a contaminant. And then I'm also looking at these cells for confluence. How much of the field of view is covered by cells? And usually we talk about confluence in terms of percent. 25, 50, 75, 100%. Confluence would be your entire field of view is covered by cells. When you determine your confluence, we usually want to passage the cells when they're about 70–80% confluent. You don't want to go above that. Too high of a confluency generally tends to harm the cells. They don't want to be that crowded for things. So it's important to estimate your confluency and regularly keep track of it day to day, so you know when to passage your cells to keep them healthy and happy. Thanks. This is Dr. Amanda Hagee, again showing you how to check your cell confluency and for contaminants using a basic inverted microscope.